Our next speaker is Professor Daniel Segre. He is a professor of biology, biomedical engineering, and physics, and is in the I would call you a professor of bioinformatics, but the bioinformatics program here at Boston University. He studies the microbiome, and he brought to us a high throughput project to the DAMP lab, which he will talk with you about today. Welcome, Daniel.
Thank you very much. So, as Kitty was saying, we started this microbiome initiative a few years ago. This was really a grassroots effort. There were a number of labs involved in microbes, and this was a fairly new thing. In microbial communities, this is a representation of all the faculty that are part of the microbiome initiative now. It's been going on for about 5 years. A lot of students and postdocs involved, and a lot of joint projects, mostly actually related to environmental microbiology, environmental microbes, as I'll mention in a few minutes. So when this opportunity arose, some of us got together, and as I'll tell you, one of the bottlenecks in our research is really seeing who's there in microbial communities. So these are Jenny Batnagar, Sarah Davis, Wally Fulweiler, and Jeff Marlowe, involved in a combination of soil, sediment, marine microbiomes in different projects. This, I think, it captures well some of our motivation. I really think this is actually from an old T-shirt from a meeting of the ASM, American Center for Microbiology. But the idea is microbes are very powerful, of course, in terms of their metabolism. There are a lot of important applications in environmental engineering and crop production, in addition to obviously the human microbiome side, but even in terms of carbon sequestration and so on, there are a lot of really exciting projects where microbes could play a key role. So as a microbiome initiative, we devised early on an overall strategy that would fit many different projects. And I really want to highlight that in general, I think there are a lot of exciting opportunities for automation in the microbiome field. What we have been trying to do is think of ways of integrating laboratory experiments and field work. For example, taking samples from soil or forest or sea sediments, taking the microbes that are present there, do sequencing, see who is there, what they do through either 16S ribosomal RNA sequencing or metagenomic sequencing, try to get some of these microbes in the lab to grow them and build communities, synthetic communities in the lab to compare with the natural communities. And in parallel, we have a lot of effort on building models, trying to map interactions between microbes, creating what are called genome scale models, whole genome models of different organisms to try and do predictive modeling of how communities will function. But at the core of all of this is really figuring out who's there and what they do. And that's where this is often a limiting step in a lot of what we do. And we need a lot of samples, a lot of sequencing of different locations, different time points, repeats, and so on. And this is costly and time consuming. Let me tell you a little bit more. You may be familiar with this, but the steps that are required for seeing who is there in a microbial community require breaking up the cells, extracting the DNA. And I'm talking here about bacteria, but this is similar for fungal cells. And amplifying typically the 16S region of the ribosomal RNA, which is a little bit like a barcode that serves as a unique identifier of specific taxa. So by reading this part, we can see who is present in a given community sample. And because we want to do this in a high throughput way for a lot of different samples, what is essential in order to make the sequencing effective is to do barcoding. So in addition to amplifying these portions of DNA, we make unique barcodes so we can pool many samples together and do multiplexing in 96 well plates. So in a single well, we may have multiple samples. And when we do the sequencing, we can then deconvolve and get a lot of information about the composition in many, many different samples. And this is really where a lot of the time effort goes in establishing this community composition. And also, even if the cost of sequencing is one of the limiting factors, in fact, if you were to outsource the amplification and barcoding, that's about half of the price. So it's a very costly and time-consuming step. So this is where we thought, if we could utilize this opportunity for the DAMP lab, this would really help us speed up our projects and create more sequence data. So in our initial statement of work, I mean, and I'm actually by training, I'm a computational biologist, so I've never done this myself, but a lot of the people involved in this project have, and there are a lot of steps, and this is why this is very time-consuming and complicated. In addition to doing the PCR itself, the purification of the samples, quantification, normalization are all steps that can be done in 96-well plates, but are perfect for automation. And the idea was to try and use this liquid handler system. One of the examples I'm going to mention a little bit in a little bit more detail is work performed in Jenny Batnagar's lab in biology by graduate student Katie Atherton. But there are a number of projects on coral microbiome, some projects in our own lab on trying to improve carbon absorption in soil microbes. And this is the one that is actually now ready to go as one of the next steps in this project. So this is Katie, and she's been working with Jenny and in collaboration with the Hutira lab to work on microbiomes of trees in the Boston area. And the idea is that we know that areas of high heat are associated with lower amount of trees, and trees in the urban areas are dying at faster rates than in rural areas. And so this is a problem, and one of the hypothesis, which is supported already by early data collected by Katie, is that the health of these trees is highly connected to the health of the microbiomes that is present in the soil. So the idea is to try and characterize the microbiome in the soil associated with these trees and see how we can associate health of the microbiome with health of the trees. So Katie has assembled a large number of samples from about 100 trees from the Boston area. And there is a lot of details that I'm skipping here, but essentially she has about 260 samples ready to go for amplification and sequencing for a total of 792 protocols. And this is one of, I think, of the most exciting projects that will be enabled by this pipeline. So this is another depiction of the steps involved in this pipeline, including the PCR, purification, quantification, and normalization and pooling. So I'll tell you a little bit one of the way we quantify how things are going. And the main thing is really validate the runs and see there are a number of steps involved after the purification and the amplification and purification. We need to send the samples to sequencing. And ultimately, what we want to see is that the samples that we sequence now through the damp lab match what was done before in a completely independent, manually done amplification and sequencing experiment. There is so the comparison of the data themselves, seeing whether it worked, and also we want, of course, to know that this is worthwhile in terms of time that we save in the process. So the data I'm going to show you were generated in 3 steps. So Jenny and her lab had developed a sequence before several samples, including 16 samples that were chosen for resequencing with the new pipeline. This was from soil microbial samples. And in the pilot, what was done was both manual library preparation, mimicking what was done before in Jenny's lab, and then automated library preparation with the liquid handlers. So there is a total of the 16 samples across these different approaches. And right now we have finished the pipeline. The damp lab has completed the pipeline. The validation runs have been completed, and we're now ready to run new data from Katie's project. And I'll show you some example of the results. But before this, I have to tell you that one of the exciting parts for me about this whole project is that it couples very well the computational and the experimental capabilities that we have in the Microbiome Initiative. This is Michael Silverstein, a current graduate student in our lab in collaboration with Jenny Batnagar. He's a microbiome fellow, and he developed a pipeline to process the 16S data on our supercomputer and created this very user-friendly way to go straight from the raw data to amounts, of different taxa in the data. And just to clarify how well done this pipeline is, I'll say that I was the one that did the analysis in the end, and it took me 1 hour of training from Michael to get these things up and running. And I'm not familiar with the details of all these algorithms, but it's really, really well constructed. And it's a capability that is available to anyone at BU and beyond. And through this analysis, we're able to plot the correlation between the abundance of different taxa and different samples. And I want to emphasize, it's a little bit tricky to compare the raw amount of taxa from different samples sequenced at different times. So what we want to compare really is that the correlations between different taxa in a given sample are conserved across the different experiments that we did. And maybe this is best seen in the next picture that shows, in fact, these correlation matrices. So these are the different samples or different time points and different locations against each other. And you can see that there is a signature of the distance in terms of who is present in these communities across these different samples. This was the original data. This is the manual version of the DAMP lab process. And this is the automated version. And you can see by eye that these have very similar patterns. I'll show you in a second the correlation that reflects this. But what is also crucial here is that we went from about 3 hours per plate of manual work to half an hour per plate with the automated process. So this is an 88% cost in time. So it's a very, very significant cut in time. And as I was saying, this is the correlation between the original data set and the newly sequenced data. So it's really, really good, which is very promising and prompted us to now start the next round of real data. This is, I think it's actually a movie of the real process. I don't know. And this again shows in more detail with the timing. And I guess the crucial thing is really the reduction in hands-on time from 3 hours to about half an hour. I'll actually mention that there is a lot of aspects of technical aspects to which you can ask Courtney and Kristen about. I want to mention that we're very happy. We didn't know exactly how this would go. The test went really well. And I think what was very nice for me to see is that the trainees were very involved, very engaged, very excited to be part of this process. They helped design the protocols to transfer them to the DAMP lab. And I think this also is creating and will create hopefully more opportunities for active participation and training. And I think a lot of the students are eager to be involved in this. I think we did this for two specific types of amplification and sequencing processes, the 16S and the ITS for fungal cells. But we want to make, this is customizable and modular and could be applied to other microbial sequencing projects. I think sample variability was one of the challenges in terms of the steps of purification required. Actually, I'm not sure, in the end, everything worked well in terms of file tracking and so on, but these are very large files and with a lot of metadata that, needs to be propagated. I think this worked really well. I will let, the other aspects, for later discussions, but I think one of the exciting parts in terms of how this worked out for us is that there was a lot of communication between the different labs in the Microbiome Initiative, the different PIs, the students, the trainees, and the DAMP lab members. So this was extremely pleasing for us and very exciting. And I think this prompts also to think about the next steps. So I should say some of these projects have been funded and are funded by the Microbiome Initiative. So there is microbiome fellows that get their own stipend and some money for typically sequencing, in fact, that can be directly used for some of this. We're also part of the sequencer instrumentation proposal. So building on what was said earlier, it would be amazing to have, be able to go really just from samples to data within BU, open up a lot of possibility, not just in terms of cutting costs, but also being more creative about how to use the pipeline. For example, we have also our own MinION sequencer. We're trying to now design it to use it for educational purposes, to enable students to go from sample collection to sequencing to building models, making predictions, and so on. So I think there are a lot of opportunities for collaborations in the future. I think one of the things that we want to still develop is making sure that we can standardize the workflow, and different labs will have slightly different sampling requirement. For example, whether you isolate microbes from soil or from ocean may lead to slightly different densities and proportions of DNA. So these are things that will have to be worked out. I want to conclude with some insight into some of the other opportunities that I think are out there in terms of automation in microbiome science. And we tend to focus a lot on genomes and sequencing, but I think there is this new rising field of what I like to call phenomics. So in addition to measuring genotype, we want to know, which tell us who is there and what they do in terms of what is written in the genome, but we can have direct insight into what they do by measuring phenotypes. For example, looking at many different bacterial strains under many different conditions. And these are very, very laborious, complicated experiments that requires, for example, just pouring these different nutrients in different wells and then taking the different microbes, putting in different combinations and so on, some of which has been done in our lab and other labs, but not to the scale that is needed to really make transformative change. And we're already working together with other labs, including Yannis Pascalidis and others, on how to go from this high throughput phenotyping to machine learning approaches that can help us characterize microbes as functional building blocks of communities and design, engineer communities, which, by the way, could also be done with automation, making combination of different microbes. So I think we'll see a lot of these kind of things in the future. It's already happening to some extent, but I think the field is wide open. And I'll conclude by thanking all of you in the Unplugged, Katie, Doug, Courtney, Katie. I think it's been really, really wonderful to work with you and my partners in the Microbiome Initiative. And thanks all of you for listening.